get going, it gives me a tremendous pleasure to introduce Dr. Jeffery Clow. It gives me tremendous pleasure to introduce Dr. Jeffery Clow. Jeff is a product of that august institution, Washington University. He did his undergraduate at Boston College. He spent some time in research at the Dana-Farber, did his MD Ph.D. at WashU, was a wonderful resident at WashU despite an interest in hematopathology.
And being a Red Sox fan. Or an Indians fan.
And being an Indians fan. So luckily his research seems to be more successful than his baseball hopes right now. But Jeff finished with a fellowship in Hempath and has worked with Tim Ley for the last 4 years. He will be going to St. Jude's in July, I believe, and he's here to talk to us today about some work he's done on functionalization of genomics in AML. And thank you, Jeff. You wired up?
That should be good. Thanks, Hannah, for that introduction, and thanks everybody for coming to hear my talk, especially John and Dr. McKenna for dinner last night. So I wanted to talk about some of the work that I've contributed to the last couple years with my research mentor, Tim Ley. I have nothing to disclose, and if you guys have any questions, just feel free to stop me, or if you can't hear, just yell at me to tell me to speak up. So I want to try to cover a few different topics today. I want to first start off by discussing our overall approach to AML genomics at WashU, and then I wanted to briefly touch on some of our observations from our recently published sequencing study on AML through the Cancer Genome Atlas research network, where we reported the genetic alterations in 200 different adult de novo AMLs. Then I wanted to follow up on some of our efforts to understand subclonal heterogeneity in AML using targeted deep sequencing. Then at the end, I just kind of want to give a brief, what I say here, a brief glimpse into the future of clinical genomics. So the genomics of AML at WashU has really been led by my research mentor, Tim Ley, and this has been funded through a PPG through the NCI, and we just renewed it for the third time. And the success of this project has really, largely due to Tim and his foresight with clinical annotation and tissue banking. So for the last 10 plus years, we've been banking every de novo AML that comes into the hospital. So we now have over 1,000 de novo AMLs that are banked, and everybody that comes in gets consented for whole genome studies. So we started doing this 10 years ago before we could actually sequence a genome, but Tim had the foresight to write that into the consent. So everything that's been banked, we can go back and do any sort of genomic studies. All the samples are de-identified and given a UPN. Up front, we make DNA and RNA for any sort of downstream studies. And then at the time of diagnosis, or sometimes later, depending on the situation, we'll do a skin biopsy, usually right at the site of the bone marrow biopsy. And we're going to use that as our source of normal genomic DNA to make somatic calls. And then the other thing that has led to a lot of success with this project is that all of these samples are cryopreserved, so they're frozen down in DMSO, so we can easily go back and pull cryovials to do any sort of functional studies. And I would say the vast majority of our 1,000 samples have greater than 10 plus cryovials with usually 15 million cells per cryovial. So it's very easy to go back and pull vials. And then all of these patients are, there's a large database, so if you're interested in MLL rearranged leukemias, you can go back and look at how many cases we have, what types of chemotherapy they got, their outcomes, and what mutations they have. And then the second arm of this has really been the genomics, and that's been through the Genome Institute, of which Tim Ley is one of the medical directors, and through collaboration with Rick Wilson and Elaine Martis. So our workhorse has typically been whole genome sequencing, and typically we'll go for about 30x whole genome coverage, and our goal is to identify all the somatic alterations. We'll report the somatic variants in the non-repetitive portions of the genome, and that's somewhat of a moving target. And then what we then do is design a custom capture rate to target all of those variants, so we can get deep coverage, usually about 200x, and all of our sequencing is done on the Illumina platform. So I mentioned briefly earlier the TCGA study. So this was recently published in just last year, where we described the, as the title says, the genomic and epigenomic landscapes of adult de novo AML. And we looked at 200 different WashU AMLs, And here's just the kind of the breakdown of the cases that we looked at. And this is a fairly representative cross-section of adult AML. A little less than half of them have a normal karyotype, followed by some complex cytogenetics, intermediate risk cytogenetic abnormalities, these are trisomy 8s. And then, we have a smattering of our classic fusions, PML-RARA, inversion 16, AML-EDO, as well as some MLL rearrangements. And this was a fairly comprehensive genomic analysis. So of the 200 cases, we did whole exome sequencing on 150. We did whole genome sequencing on the remaining 50. All the mutations were validated. We had RNA-seq done on most of the samples. We also had small RNA-seq or microRNA sequencing done on the vast majority. SNP arrays on all the cases. Expression arrays on pretty much all the cases. And Illumina 450K methylation arrays done on the vast majority of the cases. And so, this is all published work, so I just wanted to briefly go over a few of the observations from this study. So the first one that I wanted to comment on is our finding that the majority of the mutations in AML samples are really background mutations. So this was done by Dan Link in our group, where we isolated hematopoietic stem progenitor cells, HSPCs, based on expression of 34, 38, and LIN, from healthy donors. We isolated single cells by flow, put them into 96-well plates that had a stromal cell layer as well as human cytokines, grew up those individual cells into colonies. Then we isolated the colonies, made DNA, and sequenced them by whole exome sequencing. And the purpose of this study was to see, do these quote-unquote normal hematopoietic stem progenitors have somatic mutations? And so what we did is we took donors from different range of ages. We took 2 cord blood samples as well as a donor in their 20s, 30s, 40s, 50s, and 70s. And this is whole exome sequencing. And on the y-axis are the number of mutations that were identified in each donor. So you can see very nicely that the number of somatic mutations increases with age. So as the donor in their 70s had almost 10 coding mutations in their genome. Cord blood donors had very few mutations. And then when we look back at our AML samples, these are from whole genome sequencing, we also saw that the number of mutations in an AML sample roughly correlates with age. And you can see that the slope of these lines are very similar. So our take-home note from this was that somatic mutations happen during normal aging in hematopoietic stem progenitors, and the vast majority of the mutations that we're identifying in AML samples are really just passenger mutations that are then brought forward. And we estimated that you get about 14 mutations per hematopoietic stem progenitor in genome per year. So here's the model. So in any given patient, hematopoietic stem progenitors will divide. During divisions, you get mutations. That's common. Most of the mutations that happen during the lifetime of that hematopoietic stem cell don't really lead to leukemia, but they accumulate over time. So each mutation that happens, it gets passage to its progeny, and then you capture those mutations. At some point, you have an initiating event, whether it be a translocation or a DNMT3A mutation leading here, and that gives some sort of clonal dominance to that cell. And then typically you'll have more progression mutations like a FLT3 mutation that will then give rise to AML. But you note here where we have 1, 2, 3, 4, and 5. So all the mutations that happen in the history of that hematopoietic stem progenitor are captured in the leukemia. So these are the passenger mutations that happened 1, 2, and 3, and then really the leukemia mutations are 4 and 5. So the next observation was that AML samples actually have relatively few somatic mutations compared to other malignancies. So this was done by work at the WashU Genome Institute, where they looked at all of the TCGA cancers and looked at the number of mutations per megabase. So you can see AML is the lowest over here on the left, so less than 1 mutation per megabase is a characteristic of AML. We have squamous cell lung cancer all the way over here on the right with a lot more. So that makes sense. These are smoking associated. But there really isn't a strong association with exposure. I mean, there are some exposures that lead to AML, but most of our de novo AMLs have very few mutations. So the third observation was that really only a handful of genes are actually significantly mutated in AML. So of our 200 cases, we found 260 genes that were mutated more than once. But only 23 of them were deemed to be significantly mutated. And this was somewhat of a statistical approach to look at the genome space dedicated to each gene, how frequently the mutations were present, whether or not the genes were expressed in AML. And you can see these 23 genes listed here. And these are, most of these are no surprise. The top 3, FLT3, nucleophosmin, DNMT3A. You have your IDH mutations, TET2, RUNX, and you'll recognize a lot of the other genes on this list as being AML associated, and some just kind of cancer associated in general. So the fourth observation is what I'm really going to focus on for the remainder of the talk is this concept of binary genomics. So just saying that AML sample has a FLT3 mutation or does not have a FLT3 mutation is going to be insufficient. I'm going to hopefully convince you of that over the rest of my talk. And that's really based on the observation that all cancers, and AML is no exception, are characterized by intratumor heterogeneity. So within a given tumor, you're going to have some mutations that are present in all cells, some mutations that are present in only a portion of cells. And this is from the TCGA study where we just noticed, looking at whole genome data, that you can find distinct subclones within AML. And we found these subclones by identifying clusters of mutations with similar variant allele fractions. So to go into that a little bit more, a variant allele fraction is really just an allele burden. So here's a real life example, AML 31, and I'm going to talk about this case quite a bit over the course of the talk. So on the x-axis we have our variant allele frequency or fraction. On the y-axis is just the density. So in this case, most of the mutations that we found had a variant allele fraction of 46%. So that said almost 50% of the reads at that position corresponded to the variant allele. So now, in most cancers, these are heterozygous mutations. So if a mutation is present in 50% of the reads and it's heterozygous, it implies that all of the cells in that sample have that mutation. So the closer you get to a VAF of 50, that means the closer you get to a totally pure tumor. I said that here. But you also notice that we have some mutations at a VAF of 33% and another set of mutations at around 12% here. And you can see that here in the raw data. So here's variant allele frequency here on the x-axis again. Here are now read counts. So each point is an individual variant that we found. You can see that there's a large cluster here at close to 50, and then 2 other clusters. Now this cluster close to 50 we've historically called the founding clone, because that's really, these are the mutations that originally arose in that transformed hematopoietic stem progenitor that have been passaged down. And then we have these subclonal mutations. So these subclonal mutations are present in a portion of cells. Founding clone mutations are present in all cells. And thus, subclones are derived from that founding clone. So looking at this case a little bit closer, so here's the same data that I just showed. I'm just circling founding clone, subclone 1, and subclone 2. So now this patient was a 55-year-old gentleman, normal karyotype AML, that ended up relapsing at day 500. So we actually sequenced the relapse as well. So at day 500, the leukemia burden was about 40%. So on the x-axis here, I have our de novo sequencing. On the y-axis, I have the results from our relapse sequencing. So you can see, once again, in de novo, we have this cluster of variants here in the founding clone that were about close to 50%. At relapse, they were present at a VAF of 20%. So that corresponds to 40% of the cells, which is what I set up here. Now, at presentation, we had this subclone cluster here, subclone 1 and subclone 2, which were very common. So about 60% of the cells had these mutations. At relapse, none of the cells had these mutations. That subclone was totally eradicated. Same with subclone 2. Now, what we did find at relapse were a cluster of variants down here shown in green, which we're calling subclone 3. Now, all of the cells at relapse had these founding clone mutations and these subclone 3 mutations. Those are the only ones that were present. And when we went back and looked at the de novo sample, you see how this is off the y-axis here. So you could actually find these variants in the de novo sample, but they were very, very rare. Only about 1 to 2% of the cells had these mutations. So at relapse, what we had was a minor subclone that arose. And we also did find some true relapse-specific mutations that we couldn't find in the de novo sample. So this case is really the poster child for why understanding subclonal heterogeneity is important in AML. And it becomes more relevant when you start looking at what mutations are in each subclonal cluster. So in this case, we had a DNMT3A mutation and a nucleophosmin mutation in the founding clone. So all of the cells at presentation had these mutations, and all of the cells at relapse had these 2 mutations. Subclone 1 had an IDH1 and a FLT3 D835 mutation, present at de novo sample, totally absent at relapse. Subclone 2 had a FLT3 ITD, which was also present in the de novo sample, but not at relapse. And I should note that this patient got standard 7+3 induction, so there was no FLT3 targeted agent on board here. These FLT3 alleles just disappeared at relapse. At relapse, we did find an IDH2 mutation. So once again, all of the cells at relapse had DNMT3A, nucleophosmin, this IDH2 mutation. Then we also found relapse-specific mutations in RUNX1 and P53. And so this case has pretty much every AML-associated mutation that you would care about. And you can see just how much things change over time. So what we know is that de novo AML has a founding clone and one or more subclones derived from that founding clone. And disease relapse may result in the emergence of a minor subclone. So what we really wanted to address moving forward was whether or not subclones in the de novo sample really had true unique functional properties. So our approach for this was to look at 19 different de novo AML samples that we've previously characterized by whole genome sequencing. On average, we had 450 variants in each sample. And we designed a custom capture array so we could do targeted deep sequencing to follow these subclones under different conditions. So we wanted to address some really fundamental questions. The first was regional heterogeneity. So here I wanted to address whether or not leukemia taken at the same time from the peripheral blood and the bone marrow were similar in their subclonal architecture. I also wanted to know whether or not what the relationship was between blasts and monocytes and maturing myeloid cells and lymphocytes in a given AML sample, whether or not they had similar mutations or not. And then we also wanted to see whether or not experimental models that are used to study AML how they would change the subclonal architecture of a sample. We did a lot of in vitro work, which I'm not going to talk about today. Today I'm mostly going to focus on the NSG xenotransplantation model. So before we get started with this, we wanted to address whether or not targeted deep sequencing was really a reproducible technique. So here we have a given sample, AML43, where we have two different replicates. And these are taken from different cryovials. The DNA was made at separate times. The library was generated. Capture sequencing all done months apart from each other. And you just looked at the correlation between the variants. And you can see, just in this very simple view, that targeted deep sequencing is a very reproducible approach. So our first question that we wanted to address was regional heterogeneity. And as I mentioned, this was really, is leukemia in the peripheral blood and the bone marrow the same at the time of diagnosis? So here's one of our cases. So here's our bone marrow variant allele fraction on the x-axis, peripheral blood on the y. So once again, taken at the same time. And we just wanted to address, are all the mutations that we identified in the bone marrow present in the peripheral blood, and how did the subclones look? So this was an M2 with 50% blast in the peripheral blood. And you can see, if there's perfect correlation, most of these variants are going to fall on this diagonal line. And you can see that all the mutations that we found in the founding clone in the marrow present in the peripheral blood. And the subclone down here that was present in the bone marrow at a VAF of 20% was also pretty close to that in the peripheral blood. So in this one case, the subclonal architecture is identical between the bone marrow and peripheral blood. Now the other thing that we noted is that the blast percentage in the peripheral blood was 50%. So as I mentioned earlier, these are heterozygous mutations. So the tumor burden's 50. You would expect the VAF to be 25 if these are heterozygous mutations. But you can see the VAF here is about 35. So it seemed, to us, this told us that there were cells in the peripheral blood that did not have blast morphology, but still had all of the mutations that we found in the leukemia. And I'll get to that later. Oh, sorry. So we looked at all 19 cases. I'm just showing two other ones for examples here. So here's AML31 again. This is a monocytic leukemia. And you can see very nice correlation between the variants that we found in the bone marrow and peripheral blood. Here's another case, AML43. And here you can see there are some subtle differences in the subclonal distribution. This subclone here was a little overrepresented in the peripheral blood, whereas this one was a little underrepresented in the peripheral blood. But importantly, every mutation that we found in the bone marrow we could find in the peripheral blood. And this is a busy slide. But it's all 19 cases that we looked at, all the variants. So it's about 8,000 variants on this plot. And all of them bone marrow versus peripheral blood. And you can see very nice correlation between the bone marrow and the peripheral blood. So once again, every mutation that we found in the bone marrow, we could find in the peripheral blood. And then what about, the canonical mutations that people care about with AML? So DNMT3A, FLT3, IDH, nucleophosmin, and the KRAS or the RAS mutations. And we've just compared bone marrow and peripheral blood. And you can see they look fairly similar. So in summary for this part, all subclones and variants detected in the bone marrow could be detected in the peripheral blood. And this has implications for testing, right? I mean, so for adult de novo AML, it seems like the peripheral blood would be a likely surrogate for clinical testing. Now, I don't think, this was 19 cases, so I'm sure there are going to be examples where this doesn't hold true if you do enough of them. And I definitely wouldn't substitute peripheral blood for bone marrow for any sort of post-chemotherapy setting, but for the most part up front, peripheral blood should be good enough.
Do you sort your samples before you make them?
No, yeah, I should have mentioned that. So all of this is unfractionated. So we do ACK lysis to get rid of red blood cells, and then everything else is just frozen bulk. So no phycol, no purification, just bulk. And so the next thing I wanted to address was how related are different cells within a leukemia sample. So the hematopathologist in the room will feel comfortable looking at this. So here's a basic CD45 versus side scatter. So this is all from peripheral blood. So here's a case AML 28 again. You can see lymphocytes, monocytes, blasts, maturing myeloid cells. So we then purify these by looking at CD33 expression. So the monocytes are bright for CD33 as you would expect. The blasts in this case were dim. Lymphocytes should be negative. Whenever you do this sort, you get some contaminating monocytes, so we then gated on the CD33 negative cells to enrich the lymphocytes. Then you can even go down and then look at CD3 and CD19 expression within the lymphocytes. So for all 19 cases, we did this sort. We isolated these cell populations, made genomic DNA, and then did the targeted deep sequencing. So for this case, we looked at all the different populations. So here's bone marrow versus peripheral blood again, and I already showed this data. You can see the nice correlation. Now here, these plots look a little bit different. We're using a statistical program designed by the Genome Institute called Cyclone, which tries to infer subclones based on VAFs. And once again, this was an M2 with 48% blast in the peripheral blood. So when we enrich the blast in the peripheral blood, we got what I thought was the expected result. You got an enrichment of all the variants. So here now the VAF is close to 50% because like I said, we're enriching for the blast. This subclone that we found in the bone marrow also was a little enriched as well. So that was the expected result. Now, the lymphocytes. So my original assumption would be that there would be some variants that would be shared between lymphocytes and some that would be leukemia specific. And here's the result. So you can see the vast majority of the variants are almost on the x-axis here. And these are, I feel, a byproduct of flow contamination. I made cytospins for all of these cases, and you usually get about 2 to 3% myeloid cells through that lymphocyte sort, no matter how rigid you are. You can see that there are these two variants here, and these are real variants that we did manual review on. And so these did get passaged from probably early mesoderm development all the way down into these lymphocytes. But they're very rare. There are about, I think there were 600 variants in this case, and only 2 of them really looked like they were really enriched in lymphocytes. So monocytes were the next population that we looked at. So once again, this was an M2 case. It did have a fair number of monocytes in the peripheral blood, very, very few in the marrow. And so, the question that we wanted to address was these morphologically normal monocytes, which they were, how related were they to the leukemia at the level of the genome? So here are the results. So at the level of the genome, the monocytes look identical to the blasts. Morphologically very different. All the mutations that we identified in the bone marrow were present in the monocytes. And this can't be contamination because this says 100% of the monocytes have these mutations. So what about maturing myeloid cells? So these were based on dim to intermediate CD45 expression and side scatter characteristics. These guys also express CD15, indicating that they are maturing myeloid cells. Mostly they were myelocytes and beyond. And here are the results. So these morphologically mature cells also derive from the leukemia. This picture is much more complicated than the monocytes, but you can see all of these founding clone variants are present have a VAF of a little over 40%. So most of the myeloid cells that we purified are derived from the leukemia. They can still undergo myeloid differentiation. And here is where you can really start seeing some changes in the subclonal architecture. You see all of these variants in purple and pink, which were enriched in the blast, are not present or underrepresented in these maturing myeloid cells. This subclone also drops. Then you can see all of these subclonal variants in brown that are enriched in these maturing myeloid cells. So in this case, these maturing myeloid cells derive from leukemia, but clearly a distinct subclone. So a subclone has distinct morphologic properties. And this is just a byproduct of all the sequencing that we've done. We had seen this very similar pattern with this case before when we sequenced this patient's relapse. So this patient relapsed at about day 300, if I recall. And here are the results. So here's bone marrow versus relapse, bone marrow versus the maturing myeloid cells. And these actually look very similar. So slightly different here in the subclonal distribution. At relapse, this patient only had about 10% blasts and a lot of neutrophils in their bone marrow. So in this patient had a separate branch from the founding clone that was giving rise to neutrophils. But still, all of the founding clone variants that are associated with this patient's leukemia were present in their neutrophils. So I'm going to show one more example. So this is AML 31 again. So I just wanted to show this one more time to remember all of the mutations that are present in this case. And I wanted to draw your attention to subclone 3. So once again, this was the subclone that was enriched at disease relapse. And this was a monocytic leukemia. So we did the same sort. So you can see clearly this is monocytic in origin. Gated on, monocytes with CD33 expression. We did the targeted deep sequencing. Here we're comparing peripheral blood and these sorted monocytes. And the results look very similar, which is what you would expect. The monocytes are the predominant population in that patient's leukemia. Now, when we sorted out the blasts, you can see you couldn't actually find some blasts, and I don't know if this is showing up well, but there are hour rods in those blasts. And they also were positive for CD33 and have blast morphology. And when we did the targeted sequencing for the blast, you had this curious picture, where you had enrichment in subclone 3. Once again, subclone 3 was the relapse subclone. And curiously, when this patient did relapse, the morphology of the patient's relapse was blastic rather than monocytic. So there was clear enrichment of this subclone that corresponded to blasts at disease relapse. The lymphocytes look very similar to what I've shown, so mostly contamination. So also these are a little confusing, but once again, these are now all of the cases that we looked at, all 19, just looking at the population, just so you can get kind of a global view. So when we sorted out all blasts from all 19 cases and compared blast to bone marrow, you can see most of the variants are on the axis. There are a few exceptions here. This is AML 31 that I discussed, but for the most part, blasts look just like the bone marrow. Now I'm calling these non-blast, non-lymphocytes. These are monocytes and maturing myeloid cells when we could get them. And you can see pretty much in every case, the monocytes, whether or not it was monocytic leukemia or not, all the maturing myeloid cells were all derived from the patient's leukemia. We didn't have any examples where the neutrophils were not derived from the leukemia. Yeah.
Jeff, did you study these patients when they were in complete clinical remission?
We're starting to. Yeah. Our original banking protocol gave us access to the de novo and relapse, but we originally didn't bank all remission samples. So it was just we get really about 150 AMLs a year, and it's a big endeavor. But for the last 2 or 3 years, we now bank everything.
Well, it'd be interesting to see if...
Yeah, I'm going to show some examples later on. Yeah. Yeah.
This just kind of feeds off Ross, what is the analytical sensitivity of your ability to detect mutations? If you have a faster out of genomic DNA, because that doesn't happen. Is it down to like less than 5%?
Yeah, it's definitely under 5%. For the most part, I can answer that in a few different ways. I mean, the lymphocytes, I think, give a good example. I mean, but like I said, I think those are background mutations. If you look at the skin, which is, if it's clean, most of these mutations are on the floor. Does that answer your question?
Yeah, I was just if you were to look at, I was curious if this was a way to follow people for MRD.
Yeah, we're getting there, yeah, and I'll touch on that right at the end, yeah. I would say, we usually say about 1%. But a lot of that, it's somewhat biased. If you know what mutation is there, to look for an RAE2H mutation at 1% in a patient where you knew it was there previously, you have some confidence. But to do discovery, no, we don't like to go under 5%. So, and for most of our original whole genome studies, really calling variants under 20%, a VAP of 20% is very challenging if you're just doing 30x coverage. So then shown here are the lymphocytes for all 19 cases. And I don't know if some of these are showing up or not, but you can see most of them are on the floor. There are a few gray dots here. So very rarely can you find variants that are present in the leukemia that are present in the lymphocytes at a significant percentage. Now, that we did find one case where we did find a DNMT3A RNA2H mutation at a VAF of about 8% in the case. So you can find them, but if you're interested in doing kind of sequencing studies, discovery sequencing studies, from these studies it would suggest that using lymphocytes as your source of normal genomic DNA is actually very, very adequate. So in summary of this part, so blasts are expectedly enriched in the founding clone variants and can correspond to discrete subclones. Other myeloid populations at the time of diagnosis are all derived from that patient's leukemia, and lymphocytes share only few somatic mutations. So then moving forward, we wanted to address experimental models and whether or not they would preserve subclonal architecture. So the standard in the AML field is to use NSG mice, so not-SCID gamma mice. These are deficient in B cells, T cells, and NK cells. And historically, people, what they'll do is they'll take their AML, inject it into a mouse, and these mice will get leukemia after 12 to 16 weeks. And then they do functional studies. But usually they've been extremely blind to the genomic architecture of the sample that they put into the mouse, and most importantly, the genomic architecture of the leukemia that actually grows out of the mouse. So we wanted to address whether or not subclonal architecture was preserved when you did this in a mouse. And then we also wanted to test, there's a newer NSG strain called SGM3, which is a transgenic line that expresses human IL-3, SCF, and GM-CSF, and this has been reported to improve engraftment of AML. So our standard approach is we take 1 million unfractionated cells, either from the bone marrow or peripheral blood. We've tested now more than 10 different samples. We do tail vein injections. We use unconditioned mice. We try to go into 5 to 10 mice so we can see just how similar each individual mouse is. And then we'll harvest the bone marrow from the mice at 12 to 16 weeks. We'll isolate all the human cells by flow, and then do our standard genomic DNA isolation and targeted deep sequencing. And for these cases, once again, we had about 400 variants per case that we were able to track. So for people that do these types of studies, I figured I would include this. This just shows the percent engraftment that we're getting in these mice. So here we're looking at human 45 expression in all of these animals. And here's just kind of a subset of the leukemias that we looked at. And you can see some of them get up to 80% leukemia in their bone marrow at 12 to 16 weeks. And you can see this SGM3 strain in black usually did better. We had a few cases where AML71 here, we put this leukemia into 4 NSG mice, none of them engrafted. Put it into 4 of this SGM3 strain, and all of them engrafted very well. So this SGM3 strain clearly is superior to the original NSG strain for engraftment of human AML. So what we did is we went back and, like I said, did sequencing on all of these xenografts when we had sufficient cells to go after. And I'm just going to, each case is actually a little different, but AML31 I think illustrates all of the relevant points. So this is just re-showing the previous results. So here's bone marrow versus peripheral blood for this case. And you can see we have founding clone, subclone 1, 2, and 3. And here's just the same data just shown in kind of a stacked view for just the peripheral blood. Peripheral blood was our input for this sample. So you can see founding clone, which has DNMT3A nucleophosmin. Subclone 1 in red had the IDH1 and the FLT3D835. Subclone 2 with its FLT3 ITD. Subclone 3 with the IDH2 mutation. And once again, subclone 3 was the one that emerged at relapse. So when we did the experiment, we took cells from the peripheral blood, put them into a mouse, and did sequencing. So this first, I'm just going to show one mouse first, because this illustrates a lot of the points. You can see all of the founding clone variants shown in gray were present in all of the cells that engrafted. So this animal had human leukemia cells with DNMT3A and nucleophosmin mutations, as you would expect. Now, all of the cells that grew, grew out in this mouse or expanded in this mouse, also had the IDH1 and the FLT3D835 mutations. None of the cells that engrafted in this mouse had any of these blue variants, most notably the FLT3 ITD, and none of them had the IDH2 or the other variants associated with subclone 3. So we took a very kind of oligoclonal AML, became very monoclonal when you put it into a mouse. So we did a lot more. And you can see that for these first 6 that we did, you can see a lot of them look very similar to this first mouse. All the cells have, well, there are a couple things to point out. All of the cells have the founding clone variants, as you would expect. And most of these are only a monoclonal AML with also the IDH1 and the FLT3 D835 subclone. We had this one interesting case that didn't have that none of the cells had the IDH1 and the FLT3 mutations, only had the IDH2 mutation, which was, once again, the relapse subclone. And if you look back at this, this was only about 2% of the cells that we injected into this animal. So this minor subclone clearly had an in vivo kind of dominance in this animal. We kept going and ended up doing 11 different mice for this one leukemia. And I should say, all of these mice were injected from the same cryovial on the same day, so there wasn't any sampling issue there. Now for these last 5 that we did, all of them preferentially engrafted with this very rare subclone that I'm showing in green, with characterized by the IDH2 mutation. A couple of them had some cells that had the IDH1 and the FLT3 D835, but they were rare. So you'll quickly see that none of these xenografts resemble the input leukemia. Most of them become very monoclonal, so we have very dramatic subclonal restriction when we passage human AML cells in a mouse. This becomes even more relevant when you consider what strains we used here. So these first 6 use the original NSG mouse strain. So in this strain, it seems you get preferential engraftment with this subclone shown in red. When you have human cytokines on board, IL-3, stem cell factor, and GM-CSF, you favor this rare relapse subclone. So your choice of mouse strain is really going to skew the results that you get. And this is very worrisome for people that are interested in doing xenotransplantation to test any sort of targeted agent. So if you're interested in DNMT3A or nucleophosmin, you're fine with this case. These are founding clone variants present in every xenograft. If you're interested in a FLT3 D835 specific agent, your choice of strain is really going to skew your results. These are not the dominant cells in this SGM3 strain. If you're interested in FLT3 ITD, the cells with this mutation never engrafted in this case. And then IDH2, you get preferential engraftment of that subclone in the SGM3 strain. So for AML31, we have the subclone 3, which was enriched at disease relapse. And it's important to note that all of these studies that we did are really based on the in vivo finding that at relapse, this was the cell population that came back. So that one in vivo. It was a rare subclone at presentation. It was enriched in cells with blastic features. Did have an in vitro growth advantage in data that I didn't show. And it had an in vivo growth advantage in mice, especially in the presence of these human cytokines. So it clearly had an intrinsic cell autonomous growth advantage. So in conclusions for our portion on subclones, we very convincingly showed that subclones in a de novo sample can have unique functional properties, both on kind of in vivo growth properties, in vitro growth properties, and morphology. Now, xenotransplantation specifically can produce pretty dramatic subclonal restriction. As I mentioned, all of our cases looked very similar to that. We took an oligoclonal AML. Pretty much every xenograft that grew out was monoclonal. Sometimes it was a minor subclone that grew out. You had some AMLs, like AML31, had very different patterns where each individual mouse looked completely different at the level of the genome. But the important take-home note is that what you put into a mouse is not what grows out of a mouse. Choice of immunodeficient strain can really alter your results. And in work that I didn't show, there really actually is no association between a relapse subclone and what actually grows in a mouse. So that I showed AML31 where the relapse subclone did preferentially engraft. We did about 5 other cases where the relapse subclone did not engraft. And unfortunately, I can say that we've done a lot of these, and we can't really predict which subclones are going to engraft in a mouse. There's really no association between mutational patterns. A lot of the times, the subclone that does engraft actually doesn't even have any unique coding mutations. They're all kind of non-coding mutations, so we're not sure what's going on in those cases. So in future directions, I'm going to end a little short here. I just wanted to talk about some of our kind of reviews on clinical testing. So based on everything that I showed, I think it's very clear that you need accurate detection of somatic variants with deep coverage to determine whether or not these variants are present in all of the cells in the sample or in only a portion. So are they in the founding clone or are they subclonal variants? Because really, if you're interested in targeted therapy is only going to work if you're going after a mutation that's present in all the cells of the sample. That's fairly obvious. And then for AML, with the publication of the TCGA data set, really all AML discovery sequencing is over. You're still going to find some mutations, but they're going to be very rare. So, for AML, a targeted approach now is reasonable, and that's what we've kind of moved forward worked with. And so, using AML31 as an example, if you do a targeted sequencing approach here, and we've designed a platform where we looked at all of the 260 recurrently mutated genes from our TCGA set, and you can use that, and you can find somatic mutations in pretty much every case. But it's very clear here that if you're looking at AML31, and there are 3 points here because we did triplicate libraries for capture, you can see that the DNMT3A and the nucleophosphate mutation are the most common. You can assign those to the founding clone. And then all of these other mutations are clearly subclonal. So I don't think you necessarily need to say this IDH2 mutation is in subclone 3, but you need to say that this IDH2 mutation is subclonal. It's not present in all the cells of the sample. Same with FLT3. So to get to a couple of the questions, sorry, this question mark got moved over. We wanted to address whether or not identifying mutations and tracking them in vivo could be used to anticipate outcomes. So, similar to MRD, we're kind of calling it clearance rather than MRD. So we have two different cases. At day 30, they were both morphologically negative. Both of them had 2 to 3% blast by flow and morphology. Unfortunately, this patient on top relapsed at day 90 and died at day 106. This patient on the bottom never relapsed and is in CR at day 673. Both of them got standard 7+3 induction. So when we did our targeted sequencing approach on this top case that had the early relapse, you can see the mutations that we found, IDH1, EZH2, and RUNX1. And you can clearly see that some of these mutations appear to be present in all cells. Some of these mutations are present in a subset of the cells. We did the same thing with this patient with a long first remission. We found a GATA2 mutation, also present in what looks to be all the cells, and we have some subclonal variants. Now, we banked this patient at day 30 as well. And once again, this patient up here, 2 to 3% blast by flow and morphology. At day 30, though, you can easily find all of these mutations. This IDH mutation is present at a VAF of 10%, so 20% of the cells in the sample, even though they were not morphologically blasts, had the IDH mutation. Now, for this patient with a long first remission, we couldn't really find the mutations at all at day 30. So we think, moving forward, we're still trying to establish sensitivity issues, as you mentioned, but, clearly looking at this, I would predict this patient's going to, if one of these patients is going to relapse, it's going to be this patient, and clearly that's what happened in vivo. So that's the end of my talk. Let me go through some acknowledgements, then I can take questions. So this effort is really largely been spearheaded by my research mentor, Tim Ley. A lot of the work has also been done with Dave Spencer, who's a lab medicine resident and now molecular pathology fellow who also works in Tim's lab with me. Department of Medicine has been extremely helpful, especially John DiPerzio. All the sequencing has been done at the Genome Institute by spearheaded by Rick and Elaine. And obviously all of our AML patients that dedicated their samples and our different funding sources. So with that, I'll take questions. Yeah.
How much role variation is there among neoplasms which have recurrent cytogenic AML, such as if you have a CML or a blast crisis arising with a CML, or some of the AMLs with inversion 16 or 821? Is that recurrent cytogenic AML really drive it in the fewer somatic mutation?
It's a tricky question. So, we've done a lot of work with APL, so I'll use that as an example. If you look at the number of somatic mutations in an APL sample versus normal karyotype AML, number of mutations is about the same. The number of subclones is actually a little bit higher in APL, surprisingly. We would have predicted kind of what you said, that you have a strong driver and that kind of takes over. But you still have subclones that grow out. APL has a lot of FLT3 mutations, which are subclonal, or WT1 mutations, which are subclonal. Things with an MLL rearrangement typically have fewer somatic mutations. I'm not sure that I totally believe that, though. So the one limitation that we have for calling somatic mutations is our skin biopsy. So, surprising to me that, a patient with circulating blasts, when you do a skin biopsy, you get a lot of leukemic DNA in that skin biopsy. So, we've kind of had to work out an algorithm where, any patient with counts over, I think it's 20,000, or any sort of monocytic leukemia, what we'll do is we'll go back and do another skin biopsy at day 14. And that really cleans up. So, if you have more leukemic DNA in your skin biopsy, it's going to hinder your ability to make somatic calls. I think once we correct all of that, I think the number of somatic mutations is not going to be that different. And not subclones either. Yeah.
Did you study the erythroid cells and the main karyocytes?
No, we didn't go there. Yeah. Yeah, I mean, to do these types of studies, you need to get sufficient number of cells. So it's somewhat limited by that. I mean, you could probably now go back and do, we try not to do whole genome amplification because that can skew results sometimes. But yeah, it would be interesting to look at.
Have your studies yet resulted in any modification of therapy for any of the patients?
Nope. Now, AML therapy hasn't changed in 30 years. It's still 7+3. We hope moving forward, but a lot of these, the problem is a lot of these targeted agents are going after variants which are in subclones. So, at least John DiPerzio at WashU is not really interested in FLT3 a whole lot, just because a lot of times it's subclonal. I mean, they'll use some of these agents in relapse settings when all else has failed, but as upfront agents, it's still 7+3. Or if the patient's old, they go to decitabine or some sort of hypomethylating agent. Yeah.
If I understand the data that you showed correctly, you looked at in something that you showed earlier. You looked at various peripheral blood subsets
Yeah.
in patients with AML, and you found that there were things that morphologically looked like differentiated monocytes, but they carried founder mutations
Yeah.
as well as de novo mutations. What were those de novo mutations, and did you ever try to engraft those cells?
So they didn't have de novo mutations. So they had the mutations that we found in the de novo
Just small
leukemia.
populations.
Yeah, but all of those variants we could still find. We didn't do discovery sequencing on the sorted monocytes. So there was, I suspect they probably would have some new mutations, but that was all based on the mutations that we first found in the leukemia. So those were the only sites that we looked at. But they were enriched. Some mutations that we found in a subset of the leukemia cells were enriched in the monocytes or underrepresented in the monocytes. And we did not try to engraft those.
The idea that those cells would differentiate, perhaps they acquired some new mutations.
Oh yeah. Yeah, I mean, there are a few possibilities. I mean, whether or not, it's just those cells don't have a complete differentiation block and they're able to go through that cycle. They probably have different epigenetic marks that allow them to escape. So, yeah, we're not really sure what's happening there.